Like Susan said, I joined the foundation 2 years ago. I'm a neuroscientist by background, but I've been working in a number of rare disease patient research-driven organizations like Lou Gehrig's disease, spinal muscular atrophy, or other neuromuscular disorders. So I've been in the rare disease for quite a while, and I developed my expertise in translational research, which I will hopefully illustrate a little bit now by talking about what is the FPWR's strategy, especially illustrating our new 5-year research strategic plan. The FPWR mission has 2 goals. The first goal is to develop new treatment, and the second goal is to improve the health and well-being of patients with PWS. That's something very important, and this is really the goal that we want to reach. So how do we do that? When you want to develop new treatments for PWS or any type of disease, there are different types of treatments that you can consider. Single treatment, that's what you hope, one treatment for everything. Multiple treatments that can be used either to address different symptoms, because most of rare disease like PWS is a syndromic, so it affects different organs and different systems. And what you want to shoot for, of course, the idea is to have a treatment that addresses the root cause of the disease. Some will talk about curing the disease. I think usually scientists are not really, there's a debate about what we mean by curing, but this is addressing the root cause. So to develop a treatment, what do you need to know? You need to know the target. What is, if you want, the enemy that you want to combat? So you need to find a target, and you've heard Teresa talked about a summary of the scientific day that was happening during Thursday. There's a number of groups that are looking really for targets of the PWS genes against which we can develop some treatment. So these are a variety of things. It can be at the level of genes. As you know, several PWS genes are missing in individuals with PWS patients. So one way to address that is how we can countervene the loss of these genes, acting at the genome itself, using some techniques to do gene editing or gene replacement. A second one is to act at the RNA level, or we can act downstream at the protein level, and there's still a lot of work that needs to be done, but for example, Teresa spoke nicely about the work that has been developed by Ryan Potts, looking at the downstream proteins of the MAGEL2, as well as Rachel Reverick. So these are really like interesting targets. Metabolites is something that is still under research. And looking also at the pathways. What are the pathways that are defectious in PWS? So we need to have work and understand what are these targets in order to develop treatment. Once we identify these targets, we are not finished. There's a whole long process that needs to be done to move from targets to treatment. And this is called the drug development path. And what I mean by drug is, any type of, it's not only small molecules, but it can be also gene therapies. So drug development or therapeutic development. The drug development process, you've heard this many times during this conference, is a long process that starts with different stages. The first one is to identify target. This is called the research and discovery. You have to define at this stage what are the therapeutic approach that you want to use. So right now, for the moment, and this can evolve in the future, we have genetic-based therapies. You've heard about gene reactivation of the maternal allele, the silenced genes that are on the maternal allele. We are also considering gene replacement, as well as pharmacotherapies. Once you have identified a target and defined a therapeutic approach, you want to test it in animal models because you don't want to test it directly in humans, and you want to test the safety as well as the efficacy. This is called the preclinical development. And then you move, once this is successful, it has been shown to be safe and efficacious in animal models, then you can start to test it in humans, and there are several phases. This has been described nicely by Teresa yesterday with 3 phases that is looking really increasingly first and most important at the safety as well as the efficacy. And then once the drug goes through all these stages and it has been shown safe and efficacious, then the regulatory agencies will approve that drug and it will be available on the market. So this drug development path is long. It takes 10 to 15 years, you know, that's an average. It costs a lot of money. It's estimated to be around, and this is for all diseases, not for rare disease only, about more than $1 billion for one drug that you want to develop. And also a huge problem that we have with that is that 90% of failures. So this is something that, you know, when you think about treatment, that's something that you need to take into account. So how do we address that? You know, what is our role as a patient research-driven organization? What can we do, you know, facing these challenges? So the FPWR approach has consisted in 2 phases. The first phase is to advance research. As I said in my first slide, you need to identify targets. For that, you need to better understand the disease at the genetic levels, at the protein, looking at different therapeutic approach. So this is what FPWR has been doing. And this is illustrating here by a fisherman, which is really putting fish lines everywhere in every direction, trying to recruit as many investigators to be interested to work in PWS, and really trying through that kind of strategy to identify some targets. And this has been successful because, as you've seen during all these last days, we have now different targets that we can work on. So build knowledge on PWS has been really the goal of the grant programs, with the 3 pillars is looking at hyperphagia, mental health, and the genetics. But as I said, you know, now we have a number of targets. As you've seen, we have a number of clinical trials undergoing. As I just said, this is very long. This is costly. There's a high % of failure. So how can we do it? What can we do the best? How can we use the money? And what is our role as patient research-driven organization? So the 5-year research strategic plan is really, the goal is how we can accelerate the therapeutic development and develop. Our really strategy is how we can develop a strategy to de-risk the drug development and accelerate the therapeutic development. And so last year, what we did is with the research team, we did a gap analysis, looking at all the different stages of the drug development and looking at all the different challenges and trying to understand what are the challenges that we can address through different programs. So that's a very proactive approach of what can we as a foundation do that others cannot do. How we can de-risk the drug development and shorten the way of developing new treatment. And this has been constructed around 2 therapeutic approaches, the genetic-based therapies as well as pharmacotherapies. Each of them have their specific challenges, but there are a lot of challenges that are common to these 2 approaches. So we looked at the drug at the discovery stage, and we developed one program that is called critical knowledge. So this is, and Teresa is going to talk a little bit more, give you more details about our program on genetics, better understand the genetics. We have another program that is neurobiology. There's still that we don't understand about the circuitry that is involved in the food behavior, even in the normal human. So we need to advance that also in PWS. We need to advance our knowledge of the circuitry as well as in humans. So we have a program for that. These programs really address also some challenges. When can we act? Is there a therapeutic window where we have the most chance that a treatment is going to work for our patients? Where do we act? If we want to address hyperphagia, what kind of structure? We talked a lot about the hypothalamus, but this is not the only structure. And how do we act? So these are the questions that have driven this strategy. We have at the discovery, we have 2 other programs that once you identify targets, you first want to test it in a cell model, in a model of PWS in a petri dish, in a dish. So for this, we are developing a new program that is called the PWS cell-based assays that is going to help modelize PWS in a cell that will help also not only better understand what are the molecular mechanisms underlying PWS, but also serve as a really interesting tool to do high-throughput screening. We have a lot of drugs that are out there. Some of them are already on the market. Others are in development. We would like to do a high-throughput screening, so screening at, in a high-throughput manner, different drugs. But for this, we need first to have cell models. So that's something that we are developing. At the preclinical stage, and I'm going to talk just after this talk about the preclinical animal network that we have started to develop a program, a very important program last year. And we have also new animal models. We need to keep developing new animal models, whether it's mouse models, rats. We are developing a rat MAGEL2 model. We are also developing a pig model. We are considering also developing a primate model of PWS. So these are different models that needs to be also developed further in order to test any interesting drugs at the preclinical stage. And then at the clinical trials, to address the challenges of clinical trials, we have launched a PWS clinical trial consortium 2 years ago. We have invited all the key opinion leaders, including the FDA, as well as academia and industry at the international level, to really discuss about how we can address these challenges. And now we are working on some of the challenges. You've heard about the outcome measures, how we can measure hyperphagia. For the moment, we are using questionnaires. It would be better to develop more objective measures. So these are all the kind of challenges that we are addressing in this consortium through really a collaboration also between different patient organizations such as FPWR, of course, that is leading that, but also with PWSA USA, IPSO, and the French Foundation, as well as all the industry partners that you've seen some of them here that are also participating, and clinicians to this effort. Jessica will talk also the importance of the PWS Global Registry that I put here to address the clinical trial challenges, but in fact it's very important at each step of the drug development path. It will help us. And then at the market, once the drug hits the market, there are several drugs that are out there for other indications than PWS, but that could be interesting for us. And so we have a clinical care program that Teresa is going to, no, in fact it's Priya. Sorry, Priya is going to talk about that. And here the idea is really to illustrate our strategy. As you know, this drug development path is very long, so all these different programs are great, they are going to address the challenges. But we need something to develop soon to really improve the health and well-being of our patients with PWS. This is why we have this clinical care program. This is really looking at drugs that are already on the market, so you don't have to go through all this process. And we can shorten the way, the drug development process, by using these drugs that are already on the market. And we have also all these different programs, our directed programs, directed by us, that are really complementary of the grant program that has been existing in FPWR since several years. That is absolutely essential, because we need to have constant innovative ideas, new approaches that are coming and feeding, if you want, this pipeline. So it's really a nice complementation between this different directed program as well as the grant program. And we are balancing long-term programs that are addressing the challenges to develop new treatments with the clinical care that is going to be a short-term goal program. So in conclusion, the strategic plan that I just showed, it's a 5-year research strategic plan that we estimate is going to cost 26 million over, in fact, it's not just the next 5 years, it's including this year. And the goal is really, this is the take-home message, is to accelerate the therapeutic development, to one, de-risk the therapeutic development, invest in all therapeutics. As you've seen, we are not putting all our eggs in one basket, but we are investing in everything that is potentially interesting, and also to balance the short-term goals with the long-term goals. The way how we do this, it's through directed research. Now we have really a proactive way of developing this program. We know what we want. We know the answer that we want to have. And we are, this rely a lot on collaboration. And I will illustrate this, and you will see different examples about that. Thank you very much. If you have any question, please ask them.
Don't be shy. If you do have questions, you're welcome to use the mics. You can take a few now, or we don't have any takers. There we go.
So, were you saying that gene therapy is already being done? Like, because we want to have hope. We want to know that there is an end, that there is a cure. We're going for a cure. So is the gene therapy already taking place, or is that where we're headed?
No, we have a program with gene-based therapies. So we have several programs that intends to reactivate the maternal gene, the silent maternal genes. So that's programs that we have as part of our strategic plan, as well as the gene replacement also. That's something that we are also interested. For example, replacing the major 2 or other key genes and see if this can reverse the phenotypes. So these are programs as well as pharmacotherapies.
Awesome. Thanks.
Yes.
Those are many of those. Okay, many of those are in the preclinical stage now, so they're at the testing stage in the animals that Nathalie talked about. And it'll take a little while before they get into kids. It has to be shown that it works. It has to be shown that it's safe. All of that still needs to happen. So those are.
Is there a time frame for that?
I'm not going to commit to a time frame because it really just depends. If there's a safety issue, then it sets everything back. I mean, if everything goes beautifully and the FDA is open to it, you're talking I would think a minimum of 5 years before it would get into testing. If there's safety issues, there will need to be workarounds, and then that will take longer. So there's several, as Nathalie mentioned, there's several strategies moving forward at the same time because we don't know which is going to be the safest strategy.
While hope for big answers like cures and eliminating hyperphagia are fabulous right this minute, right? What have we done any research to understand better what drugs help with minor symptoms or just barely helping right this minute? So, I feel like we shoot in the dark as to what drugs help with the anxiety, what drugs, and I realize each patient is individual in how they metabolize those and how they help, but have we looked more into protocols for, besides, HGH feels like it's the only standard therapy protocol for our kids that helps them right this minute. So what other existing drugs, and have we explored how those interact with PWS and have a better protocol for how they help us?
So if I understood your questions, you are asking about, are we looking at other symptoms than the hyperphagia and looking at drugs that already exist that could be of use for the other symptoms of PWS?
I mean, I think hyperphagia, but if it doesn't eliminate or even drastically reduce it, to attack maybe the hyperphagia creates anxiety, right? Yeah. So kind of go down the line and just really explore existing therapies, existing drugs, and how they can help us. So
that's a very good question, and this is exactly what we want to address with the clinical care that Teresa is going to talk about this. Every time, sorry, Priya. She is going to talk about it, but that's exactly what we are looking at also, is drugs, looking at all the different symptoms. What are the drugs that exist out there for other indications that could be of use for PWS? But what
Right, so what I was going to say, and I'm going to talk about this a little bit when I present on the registry, is that we know that there are a lot of things that families are trying, right?
Yeah.
For anxiety, families are trying all sorts of different potential pharmaceutical drugs. They're trying different behavioral therapies, and one of the best, most powerful ways that you can get that information to the community is to enroll in the registry and to fill out the surveys in the registry. Because if you have an anecdotal, this worked for me or this did not work for me, that's an N of 1, right? But if we have 500 people or 1,000 people telling us these are the things that we tried for anxiety and this worked for us, this didn't work for us, we can start aggregating all of that data and collecting everybody's story and trying to come to some consensus of what's working, what's not working, and also what are things that we need to then do a more controlled clinical study on. If there's some evidence that a particular drug might be working, that can work in concert with the clinical care program for us to then design an actual controlled, placebo-controlled study that we can then get hard quantifiable data numbers on. So this is where all of these things work together. But again, I'll talk about it some more, but the registry is a key component of that process.
So the registry asks, what medications are you using? Did they work? Did you stop them because of some issue? It gathers all that data. So if we have that from 600 people, and then we all go on Facebook and go, what do you use for this? And whoever's on Facebook at the time answers. But that's, if we have the registry, it's many people, and it can answer that better.
Hi, this is more of a personal question. You mentioned SMA. Were you a part of that solution for that rare disease? You mentioned you worked on SMA.
You mean part of the solution?
For the gene therapy that they are now receiving?
Yeah, but I don't think I would ever say I'm part of the solution. I was there, yes, okay. and this was, this was an exciting time because
we have personal friends that have benefited from that therapy, so that just really excites me. Thank you for
and it is. I mean, it is not in PWS, but every advances that we see in other type of rare disorders, such as spinal muscular atrophy, especially type 1, where babies die before the age of 2, and you see a success, whether it's in fact pharmacotherapies using oligoantisense or gene therapies. This advanced the field for everyone, So yes, it is important to look, outside also PWS, and we learned a lot also because we have common challenges, Once you devise a gene therapy, the way how you are going to administer it to humans is also a challenge. So we can learn also from other type of disease, and that's what we also follow. Yeah.
Excuse me.
Sorry.
Can you explain the logic behind the multiple animal models, the mouse and the pig and possibly the primate? Why 3 different models? How does that help?
I think I would say the first answer is that we always try to find a better model. I will discuss about this with the pecan, but we always try to find an even better model. There's advantages in mouse models versus, for example, pig, in the sense of, you can use more mouse, you can test more drugs in a mouse than you can test, the higher you are going to go with, like pigs or primates and things like this. But at the same time, it depends what you are looking at. Some bigger animal models could be much of better use, and sometimes the regulatory agencies ask for 2 different models, in fact. So that's also something that is important, finding a better model that recapitulates better the symptoms of PWS, but also a model that is physiologically closer to what you are trying to model in PWS.
Hi, thank you all for everything that you do, first of all. There's been a lot of discussion about new drugs, and we're all very excited about that. But I think I speak for most of us that we all benefit greatly from growth hormone. And as our kids get older, it's harder and harder to obtain because of FDA restrictions on that. Is there any work being done to help us obtain that as our kids get older?
Fabulous question. So, was it planned, Susan? So Priya is going to talk about that. You know, this time I got it right. Yeah, because that's something very important. So yes, so you see, we are covering everything. Okay. Okay, so I'm going to give you an example of the type of collaboration and the kind of work that we do at the preclinical stage. So here, what you want, once you have a potential, I call it drug, but it can be, any therapeutic agent, you want to test it where there is a genetic-based therapy. The next step before it enters into human and into clinical trials, you want to test it in an animal model. So there's been a number, as you know, a number of mouse models that have been developed, in fact, for PWS, and mainly for basic research purposes. As you know, there is not only one gene that is missing in PWS, but there are several genes, and it's a complicated genetics. Some genes are coding from proteins, and some other gene codings are non-protein coding genes. So one of the questions in the field was, what is the function of these genes? So one way to approach that is to delete one gene and to see what does it do in a mouse model, to have a little bit of a clue of what is the function of these genes. When we are at the preclinical stage, what ideally you want to have is a model that recapitulates the symptoms of PWS. So you know it's a good model. But what you want to know is this model going to be predictive of what is going to happen in clinical trials? That would be the ideal. So that you know that every drug that is positive in that animal models has a good chance to be positive when you are going to test in humans. And that's exactly illustrated here. So at the top of these slides, you see this is the traditional way. If you look, you have a number of potential interesting targets at the discovery stage. Then you have a little bit less at the preclinical stage, and even less entering clinical trials. And then, this is where I told you 90%, more than 90% of failures. Then you have really like one drop that represents like really very few that makes to the market. And so you have to wait 10 to 15 years, spend a lot of dollars, billions of dollars, and then you have to start all over again. So another way, and this is where I think really a foundation like us can make really a difference, because no one else is going to do this, and I can talk about this a little bit later, is to have this innovative approach, and that's what we are doing with the preclinical animal network, is once we have these different targets, and we can increase the number of targets, is really to put the stringency at the preclinical stage. So much earlier in the drug development path. And really, by really addressing different challenges that everybody talks in the literature, you will see a lot. For example, results are not reproducible, and it's not because, scientists are cheating. It's nothing like this. But, everybody's using sometimes different protocols. Reproducing results, it means for a scientist, taking the results of someone that has been published, redoing it in his lab, and taking 1 or 2 years. And with that, you are not going to be able to publish in a good journal, so it's not rewarding for the scientists, so they're not going to do it. And then the industry is very interested to have that, but they're not going to fund that. They are risk adverse, and what they want to do is to kick in, usually at the clinical trial stage. So it's only foundation like us that can really take that risk of trying to find a way to increase the stringency at the preclinical stage. And this is what this preclinical animal network program, this is the main goal of the preclinical animal network, is really to shift the risk at the preclinical stage. And so the idea is really to decrease, because if you shorten the path of this risk, basically it's killing as many drugs as you can at the preclinical stage. So you would decrease then, you know, the cost of the drug development and also the rate of failures. So the idea here is to develop and validate based on already mouse model that are existing. We are working with a working group to, first of all, select a number of animal models that we will then screen in a very standardized way. And so what we want to do also is to validate these models for all the different symptoms that are present in PWS. So that could be also one answer to the question that was asked before, is not to look only at hyperphagia and food behavior, but is going, you know, to look at sleep, metabolism, food behaviors, social behaviors, motor function, sensory function, cognitive functions, and many other behaviors that are impaired in PWS. And the idea is really to validate what are these models, because, you know, there are also conflicting results in the literature. Some describe some animal models that can be obese under certain conditions. Others said, no, it's not true. So we want to have really a better answer first, which animal model is good for what and how good it is. And then the idea is really to create a PWS preclinical drug screening platform so that when there are interesting drugs that come out of the lab or industry has, you know, drugs that are interested in, they can go through this drug screening platform, and this drug is going to be screened for each or for every behavior and tell us, you know, how good is this drug, and, you know, what is the chance of these drugs being successful at the clinical trial stage. So that's the idea of this drug screening platform. So we launched this preclinical animal network last year in August. There are different steps in the process. The first step is to select animal models. We are reconstructing some of them, and we have really, this is really a good example of relying on collaborations with the people that are on these slides. You see, it's an international effort, and I really want to thank these people because they are really donating their time. It's not rewarding for them. For the moment, you know, maybe I hope we have some publication, but it's not rewarding for them. So they are really donating this time, and that's, you know, I really want to emphasize this because that's really fantastic. We are collaborating with the MRC, that is part of the International Mouse Phenotypic Consortium. That's you know, international efforts to make mouse models for every gene that is encoding some proteins. They have a number of tools that are really interesting, so we are relying on them also to do some part of the work. And we have 3 mice under construction. That's what is currently going on. The future plan is really to put an emphasis on looking at the phenotypic characterization, looking at different tests that are using in mouse models, and really ask the question, which is a very difficult one, is how predictive are these tests for the outcome in clinical trials? So we are going to also to have another type of expertise, more like behaviorist, and to be able to help us in this effort. And then the last step is going to be doing a screening phenotypes and then validate these preclinical animal models. So this type of program is very helpful for drug development, but also for basic research, because we'll have a better idea of which animal model is good for what, and then we can also, you know, for researchers it will be good to know that it's better to you know, look at their question, doing their research on this or that animal model. This kind of program initiative has been featured in a journal that is called Biocentury Innovations, from Ideas to IND, Investigational New Drugs. That is really a journal that is for investors and biotech company and things like that. Just to illustrate that's the kind of thing, you know, that is innovative and that got attention. So the take-home message is that type of program is really meant to de-risk the therapeutic development. It's rely on creating an international collaborative preclinical drug screening platform for PWS. So also that should help accelerate, in fact, the screening of drugs. And I hope I convince you that we, FPWR, we are in a unique position to really move forward this kind of initiative that no one else can do it, not because they cannot do it, but because, like I said, you know, it's not rewarding for academic and industry recognize the importance of this type of initiative, but will not invest in that. Thank you. If you have any questions.
So I understand the risk of de-risking, the point of de-risking and moving the risk factors to earlier. Is there a chance that we're going to miss out on some drugs that could potentially pan out, but we're not going to pursue them?
There's always a risk, you know, but I think in this case we are really increasing the chance of not passing by this type of drugs, you know, because we are really looking at, you know, as I said, in this drug screening platform, even if it is a drug, for example, that a company is developing on academic for hyperphagia, we are not going to just to screen for hyperphagia. We will look at all the different behaviors. So in this sense, it could be increasing our chance to find, you know, maybe a drug that, you know, people thought would act on hyperphagia, but actually it works on something else, and it could potentially improve other type of symptoms. Yeah.
Hi. I think it's also important. I've been lucky enough to get the layman's terms of this in the past. I think it's important to recognize that just because something cures hyperphagia doesn't mean that it doesn't mess something else up. So we don't want our kids to have psychosis because they, you know, just to get the hyperphagia to go away. So the great thing about the animal models that I'm learning is that they're literally the guinea pigs, not our children. So for me, that's comforting to know that as things get screened through these animals and they are deemed safe, and then we participate in a clinical drug trial, I have a lot more faith to know that that's going to be okay. And I mean, unfortunately, that's not the norm in, you know, all of the industry, and our kids are the guinea pigs. So it's really important to recognize that no one else is doing this. No one else is going to do this for us. It's FPWR who's really making it happen. And like she said, the researchers are doing it. They're volunteering their time for it because it's just not the common thing. But yeah, thank you so much for everything that you do. I know that Lily has been in, you know, some drug trials, and it comforts me to know that that's relatively safe for her to do. So I appreciate it.